Hello and welcome back to the Biochemistry for Health Sciences channel. As we mentioned in the previous video, the human body is made up of approximately 30 trillion cells. And from your basic biology, I'm certain most of you know the basic structure of a human cell. One of the very important questions for the healthcare professional to understand is, what do our cells do? Okay, what are the activities that go on in a typical cell in our body? So what we're going to do is a sort of a science fiction project. We are going to put Quirly in a little submarine, and then we are going to shrink Quirly and the submarine down to a size of, let's say around 100 nanometers. Okay, 100 nanometers. That's the range of the COVID nano vaccine that has been administered around the world. That's about 20 times the size of an albumin, the protein that is present most abundantly in the bloodstream. So this is about 20 times, because the albumin is about, let's say, 5 or so nanometers. Okay, so we're going to inject this nano-Quirly submarine into a volunteer's bloodstream, and then we're going to direct it towards the liver. Let's look at, say, the liver cell. That's the plan. Okay, so don't worry, Quirly. Our technology is very, very reliable. We will bring you back safe and sound. So let's begin the project. Okay, we have the nano submarine with Quirly inside, a little needle, and we are ready for countdown. Okay, 3, 2, 1, injection. Okay, your camera and lights are on, Quirly, in your submarine. We can see exactly what you see on our screen here at Mission Control. You have right now a negative. This is a negatively charged force field. This is necessary because most of these cells and particles in the bloodstream are also negatively charged. So we don't want you to collide with any of the red blood cells or white blood cells or any other particles in the bloodstream. Okay, so you are inside the cavity of a blood vessel. And yes, not surprising, you are surrounded by many RBCs. Many RBCs are on, red blood cells. Not many white blood cells. There might be a white blood cell here. Probably a white blood cell, could be a platelet, could be a lipoprotein particle. But yes, many red blood cells here. Remember, red blood cells are about 1,000 times more than white blood cells. Okay, we see some sort of protein, some particle on your right, so go ahead and head towards your right. Aha, there we go. Looks like this yellowish ball that we see here. This is probably a lipoprotein. In future videos, we'll talk about lipoproteins, the lipoprotein particles. This is probably an LDL, a low density lipoprotein, very, very rich in cholesterol. And it's about maybe 30 nanometers or so, 20, 30 nanometers. Okay, we will we'll talk more about lipoproteins. We don't see a chylomicron around, a chylomicron. Those are much bigger lipoproteins, chylomicrons. We don't see any around them. That's probably because our volunteer is in a fasting state. The last time a volunteer had lunch had a meal was about 4 or 5 hours ago. So that's why we probably do not see any chylomicrons. The bluish ball here is protein, most likely albumin, because albumin is the most abundant protein in the bloodstream. Of course, there are many different proteins in the bloodstreams, your immunoglobulin G or IgG, your clotting proteins and whatnot. So many proteins. And then we see some little chemicals here, small, brown, sort of probably organic chemicals like glucose, amino acids, okay, maybe urea, maybe creatinine, and we'll talk about some of these that are medically relevant later. And then you have even smaller chemicals here, right here, for example, enhanced by the computer to give you a pinkish purple color. Probably these are maybe ions, very small chemicals, such as your electrolytes, your sodiums, your potassiums, your chloride, your H plus protons, your bicarbonate, and so on. Again, we'll talk more about this later. So a lot of activity, a lot of stuff around in the bloodstream. And of course, this is your huge red blood cell here at the bottom right. Okay, so quickly go ahead and increase your power and your force field, and let's head towards the liver sinusoid, the capillary. So let's go ahead and do that. Yep, there we go. I can see you right here. Your little submarine, your nano submarine. Okay, let's go ahead and leave the sinusoid, the capillary. There are little holes on the capillary. You can see. We'll point here. There are little holes on the surface of the capillary. We call this fenestrations. Just a fancy name for a hole. We scientists like fancy names. Chemists especially are very good in using fancy names. Fenestrations, holes on the sinusoid. These holes are pretty big in the liver cell. Okay, so they are about 100 to 150 nanometers. So that should not be a problem for your submarine, 100 nanometers, with your force field to squeeze through. Okay, just watch the Kupffer cells here. These are the cells that can eat up anything that comes into the liver. And a lot of stuff comes into the liver. All the food most of the food that you digest and gets absorbed in the small intestine comes to the liver. Okay, so a lot of stuff comes to the liver. And once in a while, a microbe or so may also enter. So your liver is well protected by these immune cells, Kupffer cells, macrophages that can eat up everything, including your submarine. Okay, so follow your GPS and go ahead and head towards the the liver cell. Okay, so now you are in the extracellular matrix space. Okay, the space outside cells, tissue space, interstitial space, different names. And you can see this is very rich in water. There's a lot of water around. We can sense that from your submarine sensors. A lot of water. And you can see these very long strings, really long fibers. These are could be proteins or proteoglycans. We call it proteoglycans are basically carbohydrates that have some proteins. Okay, very rich. These are negatively charged. They have a lot of negative charges. so negatively charged carbohydrates such as glycosaminoglycans and proteoglycans. Okay, so very water-rich, negatively charged extracellular matrix. All right, so let's go ahead and look around and enjoy the beautiful view. Okay, so you are still outside the cell. So this is you can see the plasma membrane, right? So you can see this membrane here. Let me point it out on my screen. What you're seeing is the plasma membrane. And then you see the plasma membrane is made up of amphiphilic chemicals. So this is the head, and this is the tail. Same thing, a head, tail. So you see the heads basically this, moving around. This looks like a sea. Looks like a sea with things floating inside. Again, that's what a plasma membrane is. It's not really a liquid or a solid. We call it a fluid. Okay, and you saw this earlier when you passed through the endothelial cells too. So this is the plasma membrane, very rich in lipids, including amphiphilic lipids like sphingomyelin, lecithin, which we'll talk about later. Cholesterol, quite a bit of cholesterol in here too. Okay. And then you have all kinds of proteins. All kinds of proteins. Here's a protein, there's a protein, here's a protein. Okay, all kinds of proteins on the plasma membrane. So basically the plasma membrane is a sea of lipid heads and tails with proteins embedded in them. Okay, some of these proteins you can see are not very, quite mobile. They're moving around, while other proteins are more stationary. Okay, so there are many, many different types of proteins that you can see sticking out of the plasma membrane. Just draw some arrows to show that. And then once in a while you'll see some of this, let me use a different color, a little blue. You see this, this looks like a carbohydrate chain. So some of these proteins may have, or even the lipids may have carbohydrates attached to them. Okay, so these carbohydrates, oligosaccharides, very short carbohydrates we call oligosaccharides. Okay, so there are many proteins on the plasma membrane. So some of these proteins, they are inactive. They don't do much. For example, the MHC protein, major histocompatibly complex protein. These proteins, unless the cell is infected, you don't see much activity. And then there are other proteins that are always busy working. So for example, here you see a receptor. Here is a signal receptor. And you see signals. Let me change this color here. You see a signal here binding to the receptor. Okay, this is part of signaling. And it's kind of interesting to see how the protein shape changes quite dramatically after the signal is bound. And that's how proteins work. They bind something, and when they bind that thing, the three-dimensional shape of the protein changes. So cell signaling is a very important process in the cell. This is how cells respond to the environment. So this is the environment, the outside of the cell. And so not surprising, most of the receptors are found on the surface of the cell. So once cell signaling is initiated, the signal binds to the receptor changes shape, okay, and then it triggers other proteins downstream, and eventually other protein shapes will change. When their shapes change, their functions change. And when protein functions change, then cellular activities change. The cell starts doing something different. Okay, it may, for example, start increasing synthesis of chemicals to grow larger. The cell may differentiate into a more specialized cell. The cell might divide to form new cells. Or sometimes even the cell might undergo programmed suicide, what we call apoptosis. Okay, so cell signaling can tell the cell what to do so that it responds appropriately to the environment, what's going on around it. Okay, so although most receptors are found on the membrane, interestingly, some receptors are located inside the cell. Okay, these are examples of receptors that are inside the cell, include steroid receptors, some of the hydrophobic hormones like thyroid hormone receptors, and so on. And we'll talk about cell signaling in a future video. So cell signaling is a very important activity that cells do. Okay, so you are now nearby. Yeah, there you go. There you are. Looks like you are seeing the another very, very important activity in cells, and that is transport. Transport. All kinds of chemicals go in and out of the cell and in and out of the cell organelles. Okay, so there is a lot of transport. Water, glucose, other nutrients, ions travel in and out of the cell by a variety of transport proteins that span the entire plasma membrane. So these kinds of proteins that span the whole bilayer, the whole membrane, these are called transmembrane proteins. They basically span the whole membrane. Then there are some proteins, I don't see any around here on your screen, but there are some proteins that are only attached either on the surface or at the bottom, but do not span the entire membrane. Okay, so here in front of you, you see a very interesting, a very important protein. These are the sodium-potassium pumps. Sodium-potassium pumps. In some cells, these pumps can use almost half the energy of the cell. So you remember from your basic biology or chemistry that these sodium-potassium pumps pump sodium outside the cell, in return bring potassium into the cell. Okay, so that's why outside the cell there's a lot of sodium and very little potassium, and the opposite is inside the cell. Okay, that's due to this sodium-potassium pump. And we'll talk about the sodium-potassium pumps and their importance later in future videos. But basically, these are very useful proteins because they allow the cell to bring in a lot of other chemicals using the sodium-potassium pump gradient, what we call the sodium gradient, because there's so much sodium outside and so little sodium inside. So this is a kind of basically using the energy to store some sort of a sodium gradient energy. And that energy helps bring other chemicals into the cell. And also by pumping an extra particle, the 3 sodiums are pumped out for 2 potassiums pumped in. Basically, these proteins are removing sodium particles outside. Okay, so removing particles outside is important to maintain osmolarity of the cell, because a lot of particles are being formed in the cell. So it's important to get rid of particles. And we'll talk about osmolarity later and show how that is very important, because if that doesn't happen, then water will start going in and the cells start swelling. And once the cell starts swelling, then it gets into serious trouble and eventually it dies. Okay, so these sodium-potassium pumps are very critical. And they're also very critical for maintaining the resting membrane potential for excitable cells such as neurons and muscles. Okay, and this plays a very important role in the specialized functions of neurons and muscles. So there are all kinds of transport proteins that there are all kinds of transport proteins that bring in chemicals into the cells. So for example, glucose has a glucose transport protein, which we'll talk about quite a bit later in metabolism. Okay, so now you are in a part of a cell and you can see the cell membrane is sort of shrinking. Okay, that's what's probably is going on is a process called endocytosis. This is a transport mechanism that the cell uses to take larger particles. Some chemicals are small enough that they can go through transport proteins like glucose. But then there's some particles like proteins and like your nanosubmarine, 100 nanometers, 10 nanometers, 5 nanometers. That's very difficult to go into the cell through this very small transport protein. So they are taken in by this process called endocytosis. And then the reverse is when the cell wants to get rid of large particles, then it will send it out through a process called exocytosis. Okay, so for example, if this was the beta cell of the pancreas, you would see insulin coming out here through exocytosis. In the liver cell, you see a lot of proteins such as albumin and blood clotting factors being secreted out of the cell by exocytosis. If this was a mucus cell, say in the GI tract, you would see mucus coming out of the cell, secreted out of the cell by exocytosis. If this was a neuron, you would see neurotransmitters being secreted out of the cell by the neuron through exocytosis. If this was a plasma cell, one of the immune cells, you will see large amounts of antibodies coming out of the plasma cell by exocytosis. Okay, so exocytosis sends chemicals out of the cell, chemicals that the cell has made or doesn't want, and the process of endocytosis allows larger chemicals or particles to be taken into the cells. Okay, so one of the clinical significance we'll talk about later is the taking in of LDL, how cells take in LDL through a receptor, and then there is endocytosis that takes the whole LDL particle into the cell, and then the whole LDL particle is broken down and all the components such as cholesterol and proteins in the LDL are used by the cell. Okay, and we'll see later that if you don't have the receptor or the receptor is in lower numbers or the receptor is defective, then the LDL stays in the bloodstream, and if it stays in the bloodstream, it has a lot of cholesterol. It can be oxidized and eventually lead to atherosclerosis, and people with these problems, they get heart attacks when they're very, very young. Okay, so one proof that LDL, high amounts of LDL in the bloodstream is one of the important factors for atherosclerosis, which then leads to cardiovascular disease, which is the number one killer today. Okay, so a lot of transport, endocytosis, exocytosis, pumps always means they need energy, and then transport of small molecules, small chemicals, and so on. So there are many proteins on the plasma membrane. So for example, if this cell was an enterocyte, the cell that lines your small intestine, there are even enzymes. There are enzymes on the surface that break down food. Enzymes such as disaccharidases that breaks down sugars into simpler components to be absorbed by the cell. Okay, so many types of proteins, MHC proteins, transport proteins, pumps, and then all these proteins that are involved in endocytosis and exocytosis. So a lot of transport that happens. Okay, coolly, now we are ready to enter the cell. Well, normally a chemical the size of your submarine, we would have used that process of endocytosis to enter the cell. But here what we're going to do is sort of temporarily open up the space here between the amphiphilic lipids. Okay, sort of open up, and you're going to squeeze your way through. In fact, this is how in vitro fertilization, the needle is sent through the plasma membrane of the egg. So don't worry, you're not going to hurt the cell, because as soon as you go through, these lipids will come together by the what we call the hydrophobic force. All right, so go ahead and increase your negative force field. Okay, yes, I can see the nice big red signal there. And go ahead and go through the plasma membrane. So let me remove this and let's look at what you are seeing. Okay, so what you are seeing here, you're going through the tails here. You see, this is the tails. The fatty acid tails. These are very hydrophobic tails. They're hydrophobic, don't like water at all. This is really what we mean when we say the oil part of the cell. This is one of the major oil part of the cell. Okay, and notice that you can see part of the transmembrane protein embedded in the oil part of the cell. Okay, so in the transmembrane protein, you can see that. So this is embedded oil part, and then there's some part sticking out in the cytoplasm, and then there's a part sticking out outside the cell. Okay, we are heading towards the cytoplasm. Welcome to the cytoplasm. All right, you are in the cytoplasm. Got you tracked down here. Look at all those cytoskeletal proteins. These proteins help maintain the three-dimensional shapes of the whole cell, as well as the organelles such as the nucleus and the mitochondria and so on. Okay, so these are very important proteins that maintain the three-dimensional shapes of the cell. Cytoskeletal proteins. Yes, it is a good experience, isn't it? Very, very crowded, very crowded inside the cell. Look at all the chemicals and organelles moving around. That's incredible, isn't it? It's a good saying that's a good analogy, equally. Worse than downtown New York, lunch hour. So the cell, inside the cell is a very, very crowded place. How does anything even happen? Isn't that a miracle? See, look at all these cytoskeletal, some of these cytoskeletal proteins, right? You can see some of them are being formed, some of them are being destroyed. These are also the roads and the highways for transport. Okay, these are also the roads and highways for transport. But unlike our roads, these roads are made when needed and then destroyed once they're used. Isn't that an incredible strategy? Look at all these other ions around here and chemicals. Look at the many, many chemicals, glucose, amino acids, ions, all moving about within the water of the cytoplasm. Okay, it's rich in water. And you can see these chemicals are moving in certain directions. When you have a high concentration of a chemical, it's moving down towards a lower concentration in all directions. Okay, remember this is a process called diffusion. This is a very simple chemical principle, but extremely important in biology. Okay, things move from high concentration to low concentration, diffusion. Look at do you see all those dancing heads on the cytoplasmic side? These are the heads of the amphiphilic lipids. Okay, they look like they are dancing. They're moving around. A lot of movement even with these amphiphilic lipids. It's really remarkable. Okay, move around the cytoplasm. Yeah, we see you right now. You are near a sodium-potassium pump. Look at the sodium that is being exported out and the potassium that is being taken into the cell. Okay, it's like almost like and you can see the sodium-potassium pump changing shape, just like a revolving doors of a hotel. On your left and on your left, well, actually on our left in the screen. So right here, look at this. This is strange, isn't it? Look at this protein here. It looks like a human with 2 feet. It's 1 foot, and that's the other foot. And it looks like these proteins are carrying some particles, some vesicles with stuff inside. These proteins look like very, very much like humans walking. And you can see their feet walking on these roads, these cytoskeletal protein roads or highways. And as they're walking, they're transporting things. You can see here the transport is going in one direction. And here are more of these proteins that are transporting things in another direction. That's really remarkable. It just looks so much like humans walking and transporting things throughout the cell. So a lot of transport. Things are going in and out of the nucleus, things are going in and out of the mitochondria, things are going in and out of the plasma membrane. Okay, you see a lot of transport, and you see a lot of these proteins changing shapes. Shape change, proteins changing shapes. That's how they work. So really these proteins are the humans of the cell. If the cell was a city, these proteins are really the humans. They are doing all these activities, or at least most of these activities that cells do. So really remarkable. So if you look around, you clearly will see a lot of chemicals being made, construction of chemicals, what we call anabolism. And you see a lot of destruction, catabolism. Okay, this is what we call metabolism. So there's a lot of metabolism that is going on in the cell. Chemicals are being made, chemicals are being destroyed. Well, one very important activity you are seeing is and we'll talk about this later, is the taking in of glucose and breaking down glucose by a process called glycolysis, and then the products of the breakdown, pyruvate, those chemicals are entering the mitochondria, and in the mitochondria you're going to get most of your energy. More than 95% of the energy is made right here in the mitochondria. And energy is very, very critical for cells, right? What we call ATP. The energy is extracted and trapped as a chemical called ATP. Okay, so ATP, so energy production is extremely important. Without energy, nothing is going to happen in the cell. A lot of things like construction of new molecules, cell signaling, and so on depends on energy. So one of the major goals of metabolism is the continuous production of ATP. Okay, because cells don't store ATP, they make ATP when they need it, and they use it up. Okay, so our body doesn't have much ATP stored at any one time. Again, you see a lot of these chemicals that are going to produce energy, they're entering the mitochondria. Okay, so metabolism. Right now you're seeing more catabolism here rather than synthesis, because our volunteer did not eat for some time. So her cells are more interested in making energy to survive rather than make new things. So you are now in the mitochondria. Okay, you have gone through the outer membrane. That's the outer membrane of the mitochondria, and you are seeing the inner membrane. So the mitochondria has two membranes, outer membrane and inner membrane, and you were before in the cytoplasm. So that's where you are now. So you're basically in what we call the intermembrane space. The space, all of this is important when we try to understand energy generation in cells in the future. So in the mitochondria, you can see once again all kinds of transport proteins. Here's a transport protein, here's a transport protein, various transport proteins. And then you see all kinds of other proteins such as enzymes. So the mitochondria is probably most well known for its production of ATP. So more than 95% of all the energy in a cell is made in the mitochondria. It's a very, very important organelle, very important organelles in the cell, because without energy, most activities will stop in the mitochondria. So this is where you have very important enzymes. For example, the pyruvate that was made in the cytoplasm from either glucose or something else, some other fuel, will travel into the mitochondria, and there the pyruvate will be converted eventually will be oxidized to carbon dioxide, and then out of that will come out protons, and this is done by enzymes such as pyruvate dehydrogenase complex, as well as the enzymes of the Krebs cycle. And these protons will then enter the electron transport chain and will react with oxygen. So a lot of oxygen enters the mitochondria, and protons and oxygen react, basically hydrogen and oxygen reacting to give you water and energy. And let me just erase that. And this energy is then used by this remarkable enzyme here called ATP synthase. ATP synthase. Okay, which is next to the electron or associated with the electron transport chain or the respiratory chain. So basically ADP, the raw materials, ADP and PI. PI is inorganic phosphate. These raw materials enter the cell, enter the mitochondria, and then are converted into ATP by ATP synthase. Okay, and you notice, look at this remarkable enzyme. It's actually like a motor. It's actually like a motor is moving at 100 revolutions per second. That's a remarkable speed there, 100 revolutions per second. And every revolution produces about 3 ATPs. Every revolution produces about 3 ATPs. So you can see as the motor spins, ATP is coming out, and most of this ATP is used immediately as soon as it is made. We don't store ATP, so you can see a lot of this ATP either used up in the cell, in the mitochondrium, or it is transported out into the cytoplasm. And as soon as it's transported out, it is used up. So we don't store, humans don't store ADP. So if let's say you weigh 150 pounds, you probably need about 150 to 300 pounds of ADP per day, but we have only less than 1 pound in our body at any one time. Okay, so that's the remarkable mitochondria, very important for energy. So let's get out of the mitochondria and head back towards the cytoplasm. Okay, so a lot of metabolism goes on in the cell then, right? Metabolism. So cell signaling, transport, metabolism. So many chemicals being synthesized, such as membrane lipids. These membrane lipids have to be made because many of them get destroyed over the day. A lot of RNA and proteins have to be made because they get destroyed. So we usually, the cell makes stuff, the chemical carries out its function, is destroyed, and then new ones are made. Okay, so cell signaling, transport of chemicals, metabolism are 3 of the most common activities in the cells. And of course, the qualitative and quantitative aspects of these 3 activities differ among the various cells, the various 200 different types of cells in the body. So for example, if this was the beta cell in the pancreas, like every other cell, it will break down chemicals to generate energy. But unlike other cells, it will use a lot of this energy to carry out a specialized function. Of course, it will use energy for survival. All cells need to use their energy for survival. But the beta cell in the pancreas will use quite a bit of energy to make the protein insulin and then to secrete the insulin out into the bloodstream. Okay, so if this cell was a cholinergic neuron, again, it will use some of its energy for survival to carry out the essential things to keep it alive. But then it will also use a lot of its energy to synthesize a chemical called acetylcholine and then secrete that chemical into its environment, okay, so that it can communicate with another neuron. A red blood cell precursor in the bone marrow, for example, will use some of its energy for survival and then the rest of its energy to make the protein hemoglobin. Okay, so this is what specialized cells do. Okay, so they use some of the energy for survival and then the rest of the energy for making proteins or carrying out activities for their specialized activities. Okay, that's what physiological chemistry is. Physiological chemistry, biochemistry applied to the 200 different types of cells. And then these cells use all kinds of proteins to help carry out their specialized functions. Okay, head towards the nucleus, Corey, and The nucleus is quite a big organelle. It's about 5 to 10 micrometers. Micrometer is 1,000 nanometer. And then you can go through the hole in the nucleus. That's called a nuclear pore. That's about 70 to 100 nanometers, so you can squeeze in with your high force field. Okay, and if you look around, you can see the chromosomes, the 23 pairs of chromosomes, the DNA. That's your DNA, and you can see a lot of RNA and a lot of different proteins. So right now, this liver cell is not dividing or differentiating. So, but once the liver cell is, the liver is damaged, it can start dividing. The liver cells is very, very good in dividing during liver damage or infection or something. Okay, so cell division and differentiation is for stem cells. Stem cells can differentiate into various specialized cells. So those are the other two activities that cells do. Okay, and cell division is very common in the bone marrow. In the bone marrow, new types of red blood cells are formed in the bone marrow and white blood cells very, very quickly. Okay, cell division is an important topic in medicine due to its association with cancer, wound healing, autoimmune diseases, tissue rejection, and so on. And we'll talk about this later on. Let us summarize what cells do. So let's use the liver cell as an example. So the first thing a cell has to do is to survive. And for survival, energy production, continuous energy production, that is ATP production, is essential. To generate energy, cell need fuel and oxygen. And the most important fuel is glucose, but cells could get energy from fatty acids, which are stored in the form of fat, as well as from amino acids, which are stored as proteins. But glucose and fatty acids are the two most important fuel sources. This fuel basically contains carbon, hydrogens, and oxygen. Okay, so let's look at an example. So glucose, let's bring glucose from the bloodstream from here. So glucose from the bloodstream will enter the cell. So if there is a transport protein, let's say there is a transport protein here, and the glucose will enter the cell, and it will be broken down into smaller pieces, and the carbons and the oxygens will come out as carbon dioxide, while the hydrogens will be removed. Then in the mitochondria, oxygen which comes from the bloodstream will travel, enter the mitochondria, and hydrogens and the oxygen will combine to form water, and energy is released, and this energy is used to make ATP. Okay, so the mitochondria will continuously generate ATP, and this energy will then allow the cell to survive the energy can be used for synthesis of new molecules or chemicals that are destroyed or have to be repaired, such as membrane lipids and so on. The energy is used for pumps, for pumping actions and so on. Okay, so survival, very important. All cells need to survive, therefore all cells need to continuously make energy. So survival is the first thing cells need to do, and that is they need to make sufficient energy so that they can carry out their necessary activities going to survive. Then the second thing that cells do is specialized functions, specialized activities or functions. So for example, the red blood cell's specialized function is to carry oxygen. Similarly, the liver cell has specialized functions. So for example, after a meal, there is a lot of glucose from the diet in the bloodstream, and that will cause the beta cell in the pancreas to secrete insulin, the signal insulin. There's a lot of insulin in the bloodstream, and this insulin will come and bind to the insulin receptor on the liver cell, and the insulin receptor then changes shape and starts a cell signaling pathway, and the end result of this pathway is the liver cell's metabolism changes. It starts making chemicals. It starts synthesizing glycogen. It starts making proteins. It starts making lipids. Okay, so everything necessary for the transport and the synthesis of all these molecules will increase. Okay, so that's one of the specialized functions of the liver cell, the hepatocyte, is after a meal it starts synthesizing many chemicals. Okay, so the specialized function for the liver cell after a meal is to do a lot of synthesis. What about when you are sleeping, say 2-3 o'clock in the morning? You have not eaten anything since your last dinner, and your blood glucose is very low, has gone down. So now the pancreatic cells, the alpha cells, secrete glucagon, the hormone, the signal called glucagon, and this glucagon will now bind to glucagon receptors on the liver cells. That will start a cell signaling pathway, and now the metabolism of the liver cell changes. Now instead of synthesis, it starts breaking things down. So one of the most important specialized function of the liver is to maintain blood glucose. Maintain glucose between that 60 to 110 milligrams per deciliter or so. And that's very essential to the brain. And we'll talk about this later under metabolism. So glucagon signaling will cause the glycogen in the liver cell to break down into glucose, and this glucose will come out of the glucose transport protein and enter the blood vessel, and thereby maintain the concentration of glucose in your bloodstream greater than 50 milligrams per deciliter that is required for the brain. Okay, so that's a specialized function during starvation periods when your blood glucose is low. So the activities cells do will include metabolism, and here energy metabolism is very important. Metabolism, and associated closely with metabolism is transport. And then we have cell signaling. That's how cells know what to do to help the body. So these are the three major activities that we saw in the liver cell. And then there is, of course, cell division. In case the liver gets damaged, the cell can actually start dividing. And then there is apoptosis. In case the cell is infected, it can basically commit suicide or undergo programmed cell death. Okay, very important for the body to survive. And if it was a stem cell, of course in this case it's a hepatocyte, it's an adult liver cell, it's not a stem cell, but there are stem cells that can differentiate. Okay, differentiate. So stem cells, especially in the bone marrow, in an adult, the bone marrow has many stem cells. For example, the red blood cell precursor stem cells that can become red blood cells. Differentiation or specialization. And this is especially very important when you were zygote being transferred to a fetus and then growing from there on. Okay, so these are the things that cells do. Metabolism, transport, cell signaling, a lot of that happens. Sometimes division, sometimes apoptosis, and in case of stem cells, differentiation. So that's what cells do. Okay, I think I think we have quite a bit of information, the relevant information that's good for healthcare professionals about what cells do. So I guess mission is complete. So we are going to get you out of the body and bring you back into the needle, and then we are going to use our nano sizer to bring you back to your regular size. Okay, and there we go. And now we are going to give you a free, all expenses paid trip to Florida for going through this ordeal. Yes, enjoy yourself under the palm tree in Florida. Yeah, you are right, Claudius. Isn't that true? It is a miracle how one cell works, leave alone 30 trillion cells working together to keep us alive. Very wise. I will never ask for more. Yes, that health is the best gift of all. I'm glad this trip made you appreciate that and enjoy the moment. Until next time, goodbye.